Okay, so welcome back to this next video in which we are discussing protein folding and protein folding diseases. Okay, so in the previous video what we discussed is the basics of protein folding. We discussed that the Amundsen experiment demonstrated that the native conformation of a protein is determined purely by its primary amino acid sequence. Okay, so all of the information required to determine a protein's final native conformation is determined in that primary sequence of amino acids. Okay, right, then what we did is we talked about the Levin-File paradox, and we talked about the resolution to the Levin-File paradox, which is that proteins undergo these gradual conformational changes as they fold, and as they do so, they are effectively moving down this free energy funnel, and the conformational space that they are then capable of exploring is gradually getting smaller and smaller and smaller until it narrows down to just this single native conformation. Okay, right. What we now want to turn our attention to in this video is the mechanisms that cells have to help proteins fold. Okay, so we're going to turn our attention to the chaperones. Okay, so the chaperones is a huge, great family of proteins, okay, which are all very, very different in structure and all actually have very different functions. Okay, the only linking factor that links all the chaperones together is that they're all involved in helping proteins to fold. That's the only thing that links all of the chaperones together. Okay, so we are now going to look at two major families of chaperones. Okay, the first one that we're going to look at is the heat shock protein of 70 kilodalton family, the HSP70 family. Okay, and then what we're going to talk about is chaperonins. Okay, that's the second family we're going to look at, but there are other families that we're not going to talk about. We're going to talk about the two main families of proteins which are involved in helping proteins to fold. Okay, so we're going to start off then with this HSP70 family. Okay, now we are going to study the HSP70 family as it is in eukaryotic cells. Okay, there are very analogous proteins in bacterial cells, it's just that they're called different things. Okay, so the names that we're going to see here are going to be in eukaryotic cells. Confusingly, when we go on to discuss chaperonins, we're going to discuss a prokaryotic system of chaperonins, and then we'll later say there's an analogous thing in humans, and it involves a protein called this, okay? Because it's better understood in the prokaryotic cells in the case of chaperonins, okay? But for the HSP70 proteins, we're going to see this in eukaryotic cells. Okay, so firstly, the naming then of this family, HSP70 stands for heat shock protein. Okay, so that's the HSP, and then it's of 70 kilodaltons. That's where the that's where the 70 comes from, kilodaltons. Okay, so roughly the proteins within this HSP70 family of proteins, and I want to stress it's not just one protein, there isn't just one HSP70 protein in humans, there are multiple HSP70 proteins in humans, so it's an entire family of proteins. But the proteins in this family are roughly 70 kilodaltons in size. Okay, so remember 1 dalton is the weight of a single proton or a single neutron. Okay, so 70 kilodaltons means these are around 70,000 times the weight of a single proton or a single neutron. Okay, right. Heat shock proteins, what does that portion of the name refer to? That refers to the fact that if you stress a cell by exposing it to high temperatures, okay, by exposing it to heat basically, these proteins are upregulated. Okay, so heat shock is what is meant by exposing a cell to extreme temperatures. Okay, and then it's a heat shock protein. It's a protein that's upregulated in heat shock. Okay, and the idea there is that in heat shock, what's going to happen is loads of proteins are going to misfold because heat, remember, is a denaturing agent, and then you're going to need more of these chaperone proteins to help those proteins refold. Okay, and that's why they're going to be upregulated. So that's the origin of them being called HSP70 family proteins. Okay, right. Now, these proteins are present within the cytoplasm, okay, and they are going to bind to nascent polypeptides and also misfolded polypeptides on exposed hydrophobic portions. So remember, the characteristic feature of nascent polypeptides, or indeed misfolded polypeptides, is that they're going to have regions of hydrophobic residues that are exposed to water molecules. Okay, so let's say these are hydrophobic residues here, okay, and these hydrophobic residues, remember, are the things which drive the amorphous aggregation, okay, because water molecules do not like interacting with hydrophobic residues. Okay, so these HSP70 family proteins are going to bind to polypeptides in regions where it is where you've got an exposed hydrophobic portion, basically, and this can either be a nascent polypeptide, as I've just drawn here, okay, or it could be a misfolded polypeptide. Okay, so let me now discuss the structure of an HSP70 family protein, and then we'll discuss how they're actually going to function. Okay, so let me draw it here. Okay, so like so. I'm going to split it into three main parts, but it is all one protein. Okay, like so. So this portion here that I'm drawing now, down here, is the portion that's actually going to be involved in binding to the polypeptide. Okay, like so. And then this portion up here is going to be involved in binding to ATP. So initially, this portion up here has got ATP bound to it. Okay, so this portion in orange is the portion with ATP bound to it. And then we've got these other portions down here, which are currently in the open conformation. And you might be able to imagine what these are going to do. They're going to close around the polypeptide, which is going to sit in that cleft in the middle of them. Okay, right. So this structure that I've currently shown you is the open heat shock protein 70. Okay, so this is an open HSP70, and because it's got ATP bound to it, we'll call it an open HSP70 ATP complex, if you like. Okay, now what's going to happen is when proteins are either in their nascent form or misfolded, they'll then have this region which has exposed hydrophobic residues, and this is going to bind in this site here. Okay, so this is this site where you've got these exposed hydrophobic residues. Now initially it will form a very weak interaction. So initially this bond here between the polypeptide and the heat shock protein 70 is a weak interaction. Okay, however, what's going to happen is this portion is going to come down now and close around the polypeptide, and the interaction will become much stronger. Okay, so what's now going to happen then is you're going to close this down like so. Okay, now when that portion closes down, it requires the ATP here to hydrolyze, and it also requires another protein to actually activate this conversion of the heat shock protein 70 from this open form to what's going to be the closed form. And the other protein that's involved in closing the heat shock 70 protein is what's known as heat shock 40 protein. So heat shock 40 is involved in this actual closing maneuver, okay? And when you actually close the heat shock protein 70, you're also going to hydrolyze that molecule of ATP that's bound to the heat shock protein 70. So the ATP is going to be hydrolyzed to ADP and inorganic phosphate. Okay, now I've drawn this slightly oddly because the ADP is actually going to stay bound there. So really all that's going to be given off is the phosphate group. So let's draw this now. Okay, so here is this portion that binds the ATP. It's now going to have ADP bound there because it's hydrolyzed its ATP to ADP in inorganic phosphate. So the inorganic phosphate will now have gone off. Okay, and what will now have happened is it will have changed conformation like so, so that you now have these two portions closed in to make this little hole through the middle, and now our polypeptide is going through that little hole there. Okay, and the portion that's actually bound there is this portion with the exposed hydrophobic residues. Okay, right. So this heat shock protein 70 then is binding to these proteins with exposed hydrophobic residues. It's binding to proteins that are at risk of aggregating. Okay, these are either nascent polypeptides or they are misfolded polypeptides. Okay, now heat shock protein 70 does not actually make the protein fold correctly. What its role is to stop the proteins from aggregating together. Okay, so it's not going to do anything fancy that then helps the protein to refold. All it's going to do is stop this protein from aggregating to form amorphous aggregates. Alternatively, heat shock protein 70s are actually capable of snatching proteins off amorphous aggregates. So let's say this nascent polypeptide here had formed an amorphous aggregate. It was part of an amorphous aggregate. The heat shock protein 70 could come and snatch it off the amorphous aggregate, and therefore they're involved in breaking down amorphous aggregates. Okay, but it's not going to help it fold into the correct conformation. All it does is it sequesters it from the formation of amorphous aggregates. Okay, so what's now going to happen is the heat shock protein 70 is now going to release the protein here, so that the protein has a chance to refold into the correct structure. And if it doesn't do it, what can happen is the heat shock protein 70 can bind again, and you go round this cycle until eventually the protein does succeed in actually folding back into its native conformation. Okay, so as I say, I'll stress this again, it's involved in breaking down amorphous aggregates and preventing them from forming. Okay, but it doesn't actually help the proteins to refold. It just takes them away from amorphous aggregates and then says, go on, have a go at folding, and then if you fail, I'll snatch you back up again, and then we'll have another go later. Okay, so let me show you now what's going to happen then. In order for this thing to fold, the heat shock protein 70 needs to release the protein now. Okay, so how does it release the protein? Well, what has to bind is another protein. Okay, so I'll draw this here. So another protein has to bind onto the heat shock protein 70, and this is known as the nucleotide exchange factor, so I'll put it binding up there. Okay, and what's going to happen is when the nucleotide exchange factor binds to the heat shock protein 70, what that's going to firstly trigger is the ADP to be released here. Okay, so the ADP molecule will be released from this nucleotide binding site here, okay, which is in orange, and then what's going to happen is a new fresh ATP molecule is going to come into that nucleotide binding site, and when that new fresh ATP molecule comes into that nucleotide binding site, what it will then trigger is the opening of these two portions again. Oh, and I forgot to say, this conformation of the heat shock protein 70, this is called the closed conformation of the heat shock protein 70, and because it's got ADP bound to it, we'll call it the closed HSP70 ADP complex. Okay, right, so back to this picture down here then. So here is our polypeptide still currently bound within the heat shock protein 70 here, which I'll colour in again in vivid purple. Okay, now what's going to happen is when a new molecule of ATP binds in there, that will cause this to open back up, and then you'll go back to the very weak interaction, and then the polypeptide can come out, basically. So you're going to go back, basically, to this open conformation, okay, with the ATP bound. So you'll go in a cycle, and then the polypeptide can be released. It'll have an opportunity then to fold into its native conformation. If it fails to fold into its native conformation, it will still have those hydrophobic residues visible, and then what can happen is another heat shock protein 70, potentially the exact same one, or another one in the cytoplasm, can bind to it, sequester it away from forming aggregates, and then go round this cycle until eventually it finally does fold back into its native state. Okay, so you might be wondering, well, what is the point of these? The point is merely to stop the formation of aggregates, because if they aggregate, then they're not going to fold into the correct conformation. That's what this protein is doing. It's sequestering them away from forming aggregates. And it's also, as I said earlier, actually capable of, dragging polypeptides up from amorphous aggregates and then helping them to refold into their correct native conformation. Okay, right, so that's the heat shock protein 70 family of chaperones. Okay, what I would just like to say before we move on to the chaperonins is that you do have equivalent versions of these in bacteria, and I'd just like to give you the names for the equivalent in proteins in bacteria. So heat shock protein 70 proteins in bacteria are known as DNA-K enzymes. Okay, so DNA-K enzymes. I don't quite know how they ended up being called DNA-K enzymes when they're all around proteins, but never mind. And heat shock protein 40 proteins in bacteria are known as DNA-J enzymes. Okay, so if you're ever reading something about bacteria and you hear DNA-K and DNA-J, those are the equivalent chaperones that are present in prokaryotes. But the human equivalents are heat shock protein 70 and heat shock protein 40. Okay, now you might be wondering, well, I don't care about bacteria. Why are you telling me so much about bacteria? Well, just for consistency, because when we now move on to chaperonins, we are going to be working completely in bacteria initially, and we're going to do it the other way around. We're going to start off with looking at how this works in bacteria, and then we're going to right at the end say, and there's an equivalent system in humans. Okay, so just for consistency, I'm doing it here as well, just in the opposite order. Okay, right, so now the other family of chaperones that we're going to discuss are what are known as chaperonins. Okay, and as I say, we're going to firstly discuss chaperonins in E. coli specifically, and then we will look at the equivalent proteins in humans. Okay, so we'll start with E. coli then. Now, chaperonins are going to play more of an active role in allowing polypeptides to fold. Okay, basically the structure of a chaperonin is a chamber which you lock a polypeptide inside, and then you allow it to fold inside there. The walls of the chamber undergo all sorts of conformational changes that might be helping the protein to fold inside there. And then the lid comes off and the protein comes out, hopefully in its native state. So let's describe this structure in E. coli. So the structure in E. coli is what's known as the GroEL, GroES system. Okay, so let me firstly show you these chambers then. GroEL is going to make the actual chambers. GroES is going to make the lid of the chambers. Okay, now, unfortunately it's slightly more complicated, and it's complicated by the fact that these chambers are not sitting on their own. Instead, the chambers go around in dimers, basically. So, let me draw one of these. So you have two chambers that are sitting back to back, basically, on top of one another. I'll draw this like so. So, it's a great big barrel structure, like so. And here is one of the chambers sitting on top here, and this is another chamber sitting on the bottom. Now, let me make sure that you understand that these are two separate chambers. If I was a little man, I could go into the entrance here, I could walk down this little chamber here, but I would get to a bottom, basically, where I could not go any further. You can't go from this chamber into this chamber, okay? This chamber has a hole on the bottom here that we can't see, but I've just sort of put it there anyway, okay, that I could go into and walk up its chamber, and then again I get to a top here and I wouldn't be able to pass. So these are two separate chambers, it's just they're stacked back to back basically on top of one another. Okay, now what makes up the walls and floors of these chambers? Well, basically it's made up of 7 separate proteins. Okay, so let me draw these here. So 1, 2, 3, 4, 5, and then 6, 7 there. Okay, so I'll extend this right down. Okay, remember these lines that I drew here, these were imaginary lines. We wouldn't really be able to see those. Those should have been in dashed lines, which is why I've connected this up to here. Okay, rather than that one there. That should have been a dashed line because we were imagining that the walls were see-through basically. Okay, right. So each one of these chambers then is made up of a heptamer, which means a 7-membered structure of GroEL proteins. So each one of these proteins that makes up a 7th of the chamber is a GroEL protein. So one of these proteins that I'm colouring in vivid purple here, this is a GroEL protein. Okay, so you've got 14 GroEL proteins making up this 2-chambered dimer structure here. Okay, now, what then does GroES do? GroES is basically a lid that you can put on top of the GroEL chambers. Okay, so again, the lid is the heptamer. Okay, so a 7-membered structure. 1, 2, 3, 4, 5, 6, 7. Okay, and each one of these proteins is a GroES protein. Okay, so this is GroES. Oh, and by the way, you might be wondering, why are these proteins called GroEL and GroES? What's the Gro for? Okay, well, they were originally discovered because they were important in the growth of bacteriophages, which are viruses that infect bacteria. Okay, so humans can get viruses, animals can get viruses, but there are a lot more bacterial cells in the world than there are human or animal cells. Okay, so the main thing that viruses infect are actually bacterial cells. Okay, so the massive great collection of viruses that attack bacteria are known as bacteriophages, and basically these viruses which infect bacterial cells, it was found that for their growth, for their replication cycles within the bacteria, these GroEL and GroES proteins were utterly essential, and that's why they were called GroEL and GroES originally. Okay, but we now know what they actually do, which is that they help proteins fold, so it now makes sense why they were so essential for the growth of the bacteriophages. Okay, right. So now let's discuss how this thing actually works. Unfortunately, it's really complicated. I would love to tell you that the nascent polypeptide just goes in and then this and this and this happens. However, unfortunately, it's complicated by the fact that there are two here. And basically, when this one is doing something, this one's doing the opposite, okay? So that's what makes this story, unfortunately, much more complicated. It would be far simpler if they were just two, if each chamber was on its own, but unfortunately they cycle in these complementary manners. Okay, so let me draw out the flow cycle of how this works. So I'm going to go over to drawing this in a much, much simpler and easier to draw picture. Okay, so what I'm now going to represent this as is like so. Here are the two chambers, okay? They've been demoted down from being represented with each one of the GroEL proteins that actually makes up their walls to just being represented like this. Here's chamber on top, here's the chamber on bottom. Okay, and we can put lids on top of these chambers. So I'm going to put a lid on the top of the bottom chamber. Now, this is a cycle, so it doesn't really ever have a beginning, but we have to have a beginning at some point to discuss it. So the beginning state that I'm going to start off with is where you have a protein in here, in this bottom chamber, that has now folded, okay, and is still currently locked in the chamber with the GroES lid on top of the chamber, okay. Now, the other thing that I need to mention is the involvement of ADP and ATP in this. Okay, so in order for the chaperonins, this GroEL, GroES system to actually work, you have to hydrolyze ATP. Okay, and each one of these GroEL proteins that makes up the heptameric chamber here has a nucleotide bound to it at many points in this cycle. Okay, now this bottom chamber has now completed the folding process for its protein. It's got a folded protein inside the chamber, and therefore all of these GroEL proteins that make up the wall of that bottom chamber have got ADP bound. Now, I don't want to have to draw out 7 ADPs around every single one of the GroEL components of the chamber wall. Okay, so I'm just going to write ADP there, but what that means is that every single one of these GroEL proteins that makes up the wall of that bottom chamber has an ADP bound. Okay, right, so that's our starting point for this story. Now, it may seem a really unnatural point to start, but as I say, the reason it's a really unnatural point to start is that it's a cycle and we have to start somewhere, but there is no obvious beginning. Okay, if you look at the top chamber, you might think that the top chamber is an obvious place to start. Okay, because look, it's empty, which is exactly where we were planning to start. That seems like a sensible place to start. Okay, but because of the way that these two operate in complementarity, this one looks really unnatural, basically. It looks a really unnatural place to start. Okay, so let's actually begin this process now. So, the first thing then that's going to happen is that the lid of this bottom chamber is going to come off and the folded protein is going to come out. At the same time, each of these 7 GroEL proteins that makes up the walls of the top chamber are going to get ATP binding to them. Okay, so what we're now going to have looks like this. Each of these top GroEL proteins has got ATP bound. The ADP molecules are still bound to the bottom GroEL proteins, but now the folded protein has left the chamber and we no longer have the GroES lid covering the entrance to the bottom chamber. Okay, then what's going to happen is these ADP molecules are going to come off these GroEL proteins of the bottom chamber, and you're also going to get an unfolded protein coming into the chamber, to the top chamber, and then a GroES lid coming over the top of the top chamber. Okay, so let me now draw this. So off comes the ADP, in comes the unfolded protein, on comes the GroES lid onto the top chamber. Okay, so here are the ATP molecules still bound to the GroEL subunits of the top chamber. We've now got our unfolded protein within the top chamber, and we've got our GroES lid on top. Okay, then what happens is the actual folding process. So what happens is all of these ATP molecules that are attached to these 7 GroEL subunits that make up the wall of this top chamber are going to be hydrolyzed down to ADP, which will remain bound, and inorganic phosphate, which will be released. Okay, so I might put a big sort of flash there. Okay, you're going to hydrolyze the ATP down to ADP, chucking off an inorganic phosphate. Okay, this hydrolysis of ATP is going to cause major conformational changes of the GroEL protein. So the way that I visualize this is this whole sort of thing vibrating and churning, okay, and it is believed to help the unfolded protein here undergo many different conformations and go down that free energy funnel towards the native conformation, basically. Okay, it's giving energy, if you like, to help it move through different conformations more rapidly. Okay, right. Then what's going to happen is that process lasts for 10 seconds. Okay, so the hydrolysis of the 7 ATP molecules takes 10 seconds. Okay, and hopefully by the end of that process, what you will now have in this top chamber is now a folded protein. Okay, and what I should have probably stressed at some point is the size of these GroES systems. They are pretty damn big. Okay, they have to be to fit whole proteins inside of them. Okay, so just in case that was confusing you, that's the answer to that question. They are big. Okay, so we've now got a folded protein inside there. The other key thing is that we've now got ADP molecules bound here. Now you will notice something. We have effectively got to the same stage of the story as we begun at, but it's now upside down. Okay, so what we're now going to do is do the exact opposite, well, the exact same thing for the bottom chamber now, and return ourselves back to where we started. Okay, so the bottom and top chamber are operating at exactly opposite times. When the top chamber is working, the bottom chamber is doing nothing. When the bottom chamber is acting, the top chamber is doing nothing, basically. Okay, and that's what complicates this story, unfortunately. Okay, so now what's going to happen? Well, we can just do exactly what we had here. What happens now? The lid of the chamber is going to come off. So the lid of the chamber is going to come off, and the folded protein is going to come out. Okay, so what you're now going to go to is having the 2 chambers like so, and you'll still have the ADP bound to these GroELs that make up the walls of the top chamber. Okay, like so. Okay, then what are you going to do? Well, you're going to bring in an unfolded protein into the bottom chamber here. You're going to bring on a GroES lid here. Oh, I missed something off. Sorry, I missed something off here. Remember, that's why it's confused me having these labels put on here. You've also got to put an ATP molecule on at that point. Okay, that was one of the changes that we had in this first transition here, that you brought on the new ATP molecules. Okay, so now what we're going to do is we're going to detach these ADP molecules just as we did here. Okay, so these are going to go off. Then we're going to bring in an unfolded protein into here and then put the GroES lid on. Okay, and then what we'll have is like so. We'll have an unfolded protein in the bottom chamber with the GroES lid on, with the ATP molecules attached to the GroEL subunits of the bottom chamber. And then finally, again, it's just this process again. The ATP molecules are going to be hydrolyzed to ADP and inorganic phosphates. The 7 inorganic phosphates are going to be released, something which I didn't actually put onto this diagram over here, but should have been there. Okay, so the 7 inorganic phosphates are released, and then when the ATP is hydrolyzed to ADP and inorganic phosphates, the GroEL proteins that make up the wall of this bottom chamber are going to be undergoing vast conformational changes, and that's going to promote conformational changes in the unfolded protein that are hopefully then going to rocket it down that free energy funnel towards the native conformation, and then we're going to go back to where we started. Okay, so that then is how the GroEL-GroES system works in E. coli. Okay, so this was what happens in E. coli then. What is the analogous system in humans? Well, the analogous system in humans only has the heat shock protein 60 proteins. Okay, so there's no GroES actually in humans. Heat shock protein 60, HSP60, the heat shock protein of 60 kilodaltons, is the equivalent of GroEL. Okay, but you have no equivalent of GroES in humans. Okay, so let me outline how this works then. Again, you build a structure exactly like we've drawn here, where you make heptamers of heat shock protein 60 this time, stacked on top of one another. But instead of having a GroES lid coming on, instead what happens is the heat shock protein 60s are capable of changing conformation so that they fold over the entrance basically to the chamber. So the heat shock protein 60s in humans contain their own inbuilt lid basically. So we have evolved from E. coli. We have got proteins that have an inbuilt lid. They don't need a separate lid. They can change conformation and cover over the entrance to the chamber. Other than that, it's the same process basically. Okay, so just to review then what this does, this helps unfolded proteins or misfolded proteins to fold into their native conformation. Okay, now occasionally you can do this entire thing and the protein that you end up with will still be unfolded, or it'll be still misfolded. Okay, it can go round the cycle again. Okay, so there's nothing to stop it going through again and trying to get to the stable conformation. Alternatively, if it tries and tries and tries and fails repeatedly, eventually it will probably be degraded. And it's the degradation pathways that we will turn our attention to in the next video.